Good morning, everyone. For the day 2, today we have with us Mr. Ankit Kumar, who will be focusing on the topic medical image analysis and AI workflows in MATLAB using machine learning and deep learning approach for the today's session. I welcome you all for the day 2 of our FDP. I also welcome our speaker, Mr. Ankit Kumar. Ankit sir, good morning.
Yes, good morning, ma'am.
We will proceed with our day 2 session, sir. Over to you.
Yeah, thanks. Good morning, everyone. We will start. Yeah, shortly sharing my screen, and then we will move ahead. Good morning, everyone. Once again. So, as day 2, yeah, once again, good morning, everyone, all the participants. Hope so, all of you, as a same speaker on the second day. So, you already have covered the first day's agenda. Okay. So, in that sequence, for the second day, so we will move ahead with the medical image processing using MATLAB and AI workflow. Okay, so there is a two-part today. It's also generalized flow. It's a generalized flow. Uh, so we will focus on that. So first part, we quickly go through the resources, whatever the resources are available in the MATLAB for the medical imaging as a specific. Yesterday, I have covered the basic image processing technique. Okay, where I have talked about the programmatical approach and the app approach. You may find out that the app approach will be our save our lots of time. Okay, in the similar way, in the day 2, we have a medical image, and specifically we focus on the medical images and that what are the things are there that we were going to talk about. Okay, so yeah, let's begin. So, as all of you know, the medical image play the nowadays. Yes, as you all of you know, yes, I'm audible. No?
Yes, sir, you are audible.
Yeah. Now see here, so medical image come from the multiple sources. You may found out here in my first screen. It may become from the multiple sources such as a MRI, CT scan, X-rays, ultrasound, PET. So engineers, scientists, or data scientists, we can say that researchers must visualize, analyze with the multi-domain images, multi-domain images such as to extract the clinical meaningful informations. Okay, so this is the maybe you saw that ultrasound images are there, visualization of the voxels, 2D and 3D images, and then of course AI come. Then you have a deep neural networks or classification, machine learning classification of cells, and all so many things are there. Okay, so in the today's session, we explore the tools and algorithm of the MATLAB provided provided for the end-to-end flow. It will be the end-to-end flow, end-to-end medical image analysis, and an AI workflow. Okay, so where we can import images, 3D visualization, segmentations, labeling, analysis of medical input data that we can do. Okay, and then there's some workflow where the resources are available. As we have a 2 hours time and my PC strength is 12 GB. Okay, so there is some system requirements are also there to handle the medical images. Okay, so we will learn how to import import data, then visualize, pre-process segmentations of the image data, and how to train, use AI models on the data. Okay, so these are the things we are going to cover today. Okay, so here we look, introduce the concept, some basic concepts, not going on that depth of that, but basic concept where you can start your research, where you can explore these things with the MATLAB. The two area I am considering as for your research as well as for your curriculum as well. Okay, so yeah, so now so we have a very flexible. So let us explore these capabilities. Let us explore these capabilities for the flexible generalized workflow. Okay, so yeah, some demos I will illustrate. So medical imaging workflow, and then pre-process them, then segment out the area of interest, label the data, how can you use the TL model for train the things. We will do that. Okay, and then a medical image. So these are the things we are going to. We are going to see the some medical image labeler and radio mix and classify some things. Okay, so I have some train model already that I am going to use because time is very limited for me. Okay, and then after that segmentations and then other things are there. Okay, so we will see that the things which I am going to cover. Okay, and then after that we will provide the reference for the further explorations. You can do it with the MATLAB. As yesterday I told you, we have a very rich documentation. So of course I highly recommend the how to use documentations for your future work. Okay, so yeah, so this is a thing. Now medical images used in heavily in the various industries, various workflow, used in the various workflow planning. Yeah, it is starting from the digital pathology, OCT, radiology, maybe the MRI, X-ray, CT scan. Okay, some radiotherapy planning, and then planning endoscopy, intervascular images. You may find out. Okay, so we use this our workflow. So medical practitioners are used for the medical images such as I already told, MRI, CT scan. So create a visual representations of internal structure of the living entity. Okay, which is used to diagnosis the or treat the various disease and their conditions. Okay, so AI, now the AI will help. Now the lifespan. Now the research is going on that the human lifespan is going to increase after the discovering of AI and medical image. And this medical image will again go for the AI for healthcare. Okay, so FDA is also approved it. There how to AI can be used for medical devices. Their guidelines are there. Okay, so these are the some basic parts under the medical images you might be know. Okay, so let us understand. So medical data containers, we have a medical volume. I will show you medical volume where you can understand the medical image data. Then dedicated registers functions. If you cannot visualize with the one image, then you can how to register two images to get the full pictures. Then we have a medical image labeler that we can use it for the our today work. Then after that, I will quickly jump to the AI workflow. Okay, so where I will start with the simple machine learning, and then I will talk about the how the machine learning and different apps are there in the MATLAB. Okay, basically I will focus on the classification part. okay, which extensively used in the medical field, okay, in the AI medical field. Okay, so I will not touch on the regression part. I will touch on the classification part where I will talk about the how to plot a confusion matrix or the simple data. Okay, some feature IDs data. I will start from the basic point of view, and then quickly I will jump to the deep learning as well. Okay, so 10 line of codes how to implement, and what are the different pre-trained networks are available in the MATLAB, how to access it, what is the role of the deep network designer, and all the things I will discuss. Okay, so then after radiomic supports are the safe histogram texture analysis. This is a thing, and then you have a Monai for the segmentation also. So if you have a, you can directly interface the Monai for the segmentation. One app is there. Okay, so you have to just download the support package. So for the segmentation, you can use the Monai and its algorithm directly in the MATLAB. Okay, so means third party community tools can also be used here. Okay, so but that for that purpose you require the GPU in your laptop, and the CUDA enable GPU you must have, so that the proper segmentation and visualization can be done. Okay, so for the Monai you can interface. I will show you how to interface that part also, and then cell post for the microscopy, how that also is there. Okay, so let us jump. This is the complete workflow. So we will start with the import the data, then visualize it, then pre-process it and register, then segmentations, labeling, training, and analyze. Okay, this is the complete flow for the any medical image workflow. If you are working with the medical image, then these are the steps you have to work. And the work in fact, I would suggest that each point is itself one day discussion. Okay, so it's just a visualization itself one day, more than one day it will require to understand fully. Then pre-process part, then training itself is a very big thing. Okay, so what are the different layers, how to train, what are the different things, then labeling also take a like that. So I will quickly summarize the things and how we can do that. Okay, so yeah, with the help of MATLAB we can do very quickly. Also, I'm able to incorporate the basic idea of the medical imaging process, so you can understand. So yeah, of course. So we will start with the imported data, then the following visualization one tool is there. You can visualize it. Yesterday I have color mapping was I have shown you, but that is for the 2D 2D type of the images. But medical images are the generally 3D, and slices are there. Okay, so like that. Then yeah, segmentations and all the things we will do. And when you combine label plus segment, then it's become the ground truth, that is human validations. Okay, so this is the part. Okay. Now we'll see one by one. We'll see the yeah, this is the command. So if you just pass this command, medical volume, and of course this one, so we will see, I will tell you, then it will give you the all the information about that particular image. So medical volume, like what is the voxel size of your image, then volume geometry, then the spital units orientations. Okay, and the voxel spacings, what is the voxel spacings, normal vector, then all the information you will get. Modality, what is the modality type. Okay, so window centers and all the things. So this is the all the information you get that particular image. Okay, so we will start. Let us suppose you have some working with the TensorFlow or Cafe. Okay, so you want to bring in the MATLAB. So interoperability, import and export the deep learning framework from the PyTorch to RoNX and from RoNX to the MATLAB or some other things also we can do. Okay, so MATLAB in the latest version you directly import the TensorFlow model to the MATLAB and the visualizer things. Okay, so this interoperability is easily done the MATLAB. So there is no problem with the other software with the MATLAB. Okay, so yeah, visualize 2D, 2D plus and 3D medical images. Okay, we'll do one by one. So this is the command im play montage volume viewer and medical image labeler. Okay, so these are the four commands, or the more commands are available where you can visualize your images. So we will start from here. Okay, so we directly jump to the our MATLAB, and then I will talk about the medical image and all the things. Okay, so this is my generally I will less talk on the PPT and directly talk on the software. Okay, so I will open my MATLAB, and then we will move ahead. Okay, so there is only one code is there. I'm going to share with you quickly. Just allow me to give you just a minute where I have kept just a minute. So just a minute. Yeah, if you have any question, then ask. Meanwhile, I am sharing the code today code with you, so that you can also do along with me. Okay, so simply MATLAB online you can open and then simply copy the folder and just previous one yesterday whatever the things we have done similarly today also. So very small codes are there, and other things I will share the link directly on the chat box from where you can get the different types of files. Yeah, for a while I'm on sharing my panel, and then I come back. Just a minute, I am sharing the code. Today code is a very simple. Just a minute. Yeah. So yeah, you will get a code. Just all the codes I'm sharing. If you have any doubt, please put in the chat box. Yeah, just a minute. Yeah, just a minute. Yeah. Because what happened, today's file is not able to open. Yeah, so under that circumstances, so basically the medical images still complete folder I am sharing with you. So you can understand the codes. File is ready. Just take this folder for your reference. Everyone open your MATLAB account. Okay, so yeah, MathWorks account, everybody. In that you will find the one link. Yes. The code. And everyone open the MATLAB meanwhile. Everyone open the MATLAB meanwhile. Uh,
So your screen is not visible.
yeah, yes. Screen is not visible. I'm sharing the file.
Okay, sir. No.
Yes. Just a minute. Just I'm sharing the one file with you so that you can do the some hands-on exercise as well along with me. Just need to got it. Just actually what happened. Yeah, so everybody open the MATLAB. Yeah, everybody open the MATLAB. Meanwhile, just so yeah, link has been created. So yeah, just yes, everybody. Yeah. Mhm. Yeah. So you will get a code. Just so so Yeah, everyone. Yes, in a longer time. Sorry for that. Yeah. So yeah, this is the because and soon we have to give now. Send it. So now again, everyone, this is a three folder I am sharing with you so that we start the hands-on. Hello, hello, madam. I have shared the code with you, so kindly check and share on the group. You
Yes,
are
sir.
welcome.
Yes, yes, sir.
Please, please. Yeah. So welcome again back. So the codes are yeah, codes are I have shared. You will get a code shortly. Okay. And then yeah. So this is my code. Yeah, everybody open the code. Okay. So first code biomedical example.mlx, the first code we are going to see, and then see the what is the difference between the our normal image processing technique and the other thing. So we will quickly cover the same. Okay, so yeah, so this is the first thing we are going to do. We are here. We are just to quickly recap. This is your MATLAB
Sir,
drive.
your screen is not visible.
Yes, yes, just a minute. Yeah, yeah. Thanks for the informing me. Uh, Where is it? Yes. Not it won't be hope so. Okay. Yeah, everyone, this is the code I have shared. You will get in the yeah. So now this is the simple. I am just using the how to access the DICOM images and all the things. Okay, so I1 is equal to IM read. I am just passing this command. So everyone just is I have written very simple. So now see that everyone able to access the code. Just confirm me. Everyone able to access the code. If you are able to access the code, then we will move ahead. So first thing first, let us impose a DICOM image. It's a very popular image. Let us understand this image. I have written here. I have written here. Yeah. So everybody, chat box, you will have a all the code. Okay, so just click on that. After clicking, you will direct you to the MATLAB drive. Okay, if you open your MATLAB account, then just copy in on your own folder. That's all. That you have to do it, and or you will use the MATLAB online also. Okay, so this is the MLX file. You have to open this. Okay, biomedical example underscore example, the first myth, and let us understand some basic codes. So this is a DICOM image. Okay, so I have used the I1 variable name, and then I pass an inbuilt function im read, and let us see that whether the im read function is equally applicable for the medical images or not. I have taken this ct yeah, so yeah, let us see that. Yeah, and see if error is coming or not. Of course, if im read is applicable for the jpg and other normal images, or it equally applicable for the dicom images or not, let us see. As I know, it will give me the error. Okay, because it is not applicable, so error will tells about the just see here, you also run. So yeah, I will change the, I will change here. So now see here, the invalid expression, check for missing the extra character. Okay, so im read function is not applicable for the dicom files. Okay, so dcm file dot dcm file is not applicable for im read. So for that purpose, we have a, now you will get, everybody I think will get this is error. Okay, then I have explained you why this error is coming. Okay, so we cannot apply the im read function for the dicom type of the images. Okay, now everyone got an error. Yes, now jump to the next second line of the code. I have created another variable name I2, and then I have passed a command dicom read. Okay, and now see that where the again I am getting the same error, or it image get loaded. Okay, okay, so just see here, and now I am using the command im show I2. If I use the im show command, the image will be not properly displayed. Okay, so to show the image, I will pass a command im say. Okay, so just run this command, this mlx file. Use the MATLAB online. I highly recommend. Now see that here your image is there. Okay, now see here, but image is not properly displayed. Okay, image is not properly displayed. If you use the command, I'm just differentiating the im whatever the image processing. Yesterday we have learned some basic command. We have learned just around 10 to 15 commands only. Okay, so I'm just differentiating the different command. If I just simply search that the what exactly the im read command, and then I have told you just select the inbuilt command and go for the help. Okay, the help command. If shortly it will be pop up this short information, whether you have this type of the information. If you want the further analysis, to click here, learn more. It will open another folder, and you will get all the details how to read the DICOM images. Okay, so this is the thing. So DICOM read file name. Okay, so how to read different DICOM images, and here you will get all the informations. Okay, mlx, yeah, you have ultrasounds, different types of ultrasounds, then the DICOM file to display name, and all the things you will get it from here. Okay, so we have to pass a command DICOM read to read the medical images. So yeah, so now come here, to understand that DICOM read will be explained. Now second thing, so but here the image is not correctly displayed. Okay, so im show also not applicable. Im show is also not applicable for the medical images. So for that purpose, you have to use im this command you have to use and click here and you will get this image. Okay, so yeah, so this is the proper image of the. So I don't know about what is exactly the image is about and then what is the information it contain. Okay, so yeah, so this is the image, biomedical image. Okay, and this is the not properly display image where I have used the IM show function, and before that I have used the IM read function, which is not allowed, not applicable for the medical image, and DICOM read I have applied to understand. That's all. That much I have explained. Okay, hope so all of you run this. Now I don't know about the informations. Now I will pass a command DICOM info. Click on the DICOM info, and now from here you will able to get all the informations where this image has been stored in local drive, or the what is the file size, what is the format, okay, what is the depth, width and bit depth, what is the color type, okay, all the informations you will get. Okay, if you
Ankit
go
sir,
for
sorry to interrupt,
yes,
but
please.
the folder that you have shared, they do not contain this file, biomedical YouTube example. You have shared DL day2
Yes.
folder,
Uh-huh.
and I can see on your screen it is day2 demo folder. Can you please check once again?
Madam, what is the folder is showing over there?
DL
What
day2.
under that
Under that there is AlexNet and GoogleNet file.
okay, 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 fine, fine. Here is a day to day. Is there day to demo first biomedical underscore example?
So you have not shared this folder.
Okay, fine. Okay, okay. Okay, DL day 2 I have shared. DL day 2.
Yes, sir.
Okay. Okay, that is also required. That folder is required in the after 1 hour. Okay, fine. So this current folder again I am sharing with another. Just a minute, you will get shortly. Yeah. Share. Create a link. So very easy with the MATLAB, right? Create a link. Copy the link. Without subject I'm sending. Just check it, man. Day two demo. Yeah,
Yes,
everyone.
sir. Yes.
Yes, yes. Share it on the chat box. Okay, I can also share it. Okay, fine. Okay, fine. So you will get that file folder is also required for the our sessions. Okay, so I will little bit take some time. Okay, I'm not able to conclude. Yeah, so just see here, everybody take a time, load the image, and go through these corrections. So you have a better understanding. DICOM image, IM read, and all these functions. Okay.